you put pressure on these cells, they will develop in the right way. However, this pressure is very difficult to apply because it can be some spirally acting force. It is very difficult. It is possible. And what is probably inside, there are those piezoreceptors. So this is some kind of future. And let's go to David Julius. Discovery of this TRPV1, the first vanilloid receptor discovered, and then the others. What is the meaning of that? How he got a success? This is quite, in my opinion, important slide, because first they created a library of million DNA fragments corresponding to gene that were known to be expressed in the sensory neurons. Then they hypothesized that the library would include a DNA fragment encoding the protein capable of reacting to capsaicin. So at the very beginning, the target was quite different. They express individual gene from his collection in cultured cell so that normally do not react to capsaicin. After a very tremendous work, a single gene was identified that was able to make cells capsaicin sensitive. Then further experiment revealed that the identified gene encoded a novel ion channel, later named TRPV1. Then they investigated the protein's ability to respond to heat, and after that they realized that they had discovered a heat-sensing receptor on such a way. Look at how difficult way it was. So there are a lot of, for instance, paper for that. This is the original paper of the, as you can see, this Adam Papatougian. It's very difficult, this name for Papatougian. And the next comment. You know. As a scientist, we are told to publish to get points. I checked both of them. They published at about 60 papers. Not too much. As the first authors, they published not than 10 or several. So. But what was the important was what was published, not how much. This is the issue. So the quality, not quantity. And look at that, normal nerve endings and lot of channels. Some of them, they are sensitive, they can open or close within some kind of temperature range. Some of those channels, like TRPA1, are in fact these thermoreceptors. Look at there's a huge cocktail of that, and this is a place also for capsaicin and other substances. Some of those channels are sensitive to some chemicals. We know this empirically. And then we've got, let's say, we've got these two channels, TRPV1, which is very active, which opens at relatively high temperature, and TRPM8, which is open, which can open at really low temperature, and both of them, under some circumstances, can open in the temperature between. So this is This is not easy story. And you can see here how they respond, for instance, to burning, cold, cool, tepid, that means medium, warm, hot, or burning hot. By the way, we've got more than two of them. We've got more, TRPV1, TRPV3, TRPV2. So they work within some kind of range of temperature. And some of the, what is their function? Modulation of the actual potential of the cell. And on that way, they can act. So, look at that. This is some kind of ligands for those channels. Some of them we know: mint, cinnamon, garlic, camphor, chili. This is capsaicin originally as an active substance in here. And you can see that those channels, each of these channels has its range of temperature in which it responds. The range is relatively narrow, but we've got a lot of channels. What more? If we've got, for instance, free nerve endings, we've got also on the skin the surface of keratinocytes. Some of those keratinocytes, they have a high expression of TRP3 and 4. And after following change of temperature, these keratinocytes releases some cytokines that influences, in turn, neurons. This is how it works. We've got here, you can see gene markers of those free nerve endings, but by the way, also for specialized tactile receptors. So our feeling of touch or pressure can be modified by temperature, but conversely, we can feel also pain on that way. Look at, see that this is not uniform expression of those channels. Some channels are quite specific, other not. Like this noxious stimuli, acidity, cannabinoids, cytokines, and so on. Once again, some of them are activated by cooling, other by heating. A lot of commonly known spices, sugar, they can simply change our perception of temperature, but also pain. Empirically, we know about it. Okay, and look at this neural pathway. This is not only the story of nerve endings. This is also a story of what is happening on the way along there. And you can see some of those channels respond to heat or to cold, but other to mechanical forces. So they are close in the profile of action with piezoreceptors, and the other to chemicals. I'm slowly going to the end. And we've got also similar channels in the class of track channels. This is not but look at that, what is written here. Activation of those channels by increasing the temperature reduces excitability of the receptors. But on the other way, activation of TRP's receptor increases excitability of thermoreceptors, but some are activated by an increase or another by a decrease in temperature. So this is not an easy model from the very beginning. We started from that channel. Finally, we've got brain. I will not tell anything about that. But look at that the beginning is at the level of nerve endings. But then we have also those channels in the whole central nervous system. This is a pathological decrease of cold-induced pain threshold perception. An example, this work like that. Activation of deactivation of those channels is one of the reasons for some pathologies. Look at the expression of these TRP channels in the central nervous system. Astrocytes, microglia, oligodendrocytes, central nervous system neurons. There's a huge expression of those channels. So, what does it mean practically? That if we try to interfere with those channels, we may get an effect. Now it's I've read today that there's some kind of hope for Alzheimer's disease using this. And the last but not least, the encoding of that information is really quite complex process. For instance, warm information, usually, if the information is transmitted because some warm channels were activated, usually it reduces threshold for pain. But on the other hand, there are also cold pathways there, and they may interfere. And this, and in normal conditions, the warm fibers, they inhibit cold fibers, and these cold fibers may decrease